इस वीडियो में हम डीएनए एक विरासती मादा है। डीएनए इज अ जेनेटिक मटेरियल, इसके बारे में पढ़ेंगे। तो चलिए शुरू करते हैं। जैसा कि हमको मालूम है कि डीएनए ये हमारे साथ एक विरासती मादा है जो मुख्तलिफ किस्म की मालूमात को एक नस्ल से दूसरी नस्ल में मुंतकिल कर देता है या मुख्तलिफ किस्म की खुसूसियात वालिदैन से बच्चों में मुंतकिल करने का जिम्मेदार है। But how we know that DNA is a genetic material? We know that protein plus DNA it produce the chromosome. So which either protein or DNA, in both of these, either both of them they are responsible for heredity or one of them. So with the help of experiment, we prove that DNA is a genetic material. There are certain properties of genetic material. We know that DNA is a molecule. To act as a genetic material, it should have the following properties. Be capable of replication from its own replica. इसका मतलब है कि डीएनए रिप्लिकेशन, एक डीएनए से दूसरे डीएनए बनने के अमल को हम डीएनए रिप्लिकेशन कहते हैं। मतलब इसमें ये प्रॉपर्टी मौजूद हो। इट शुड बी स्ट्रक्चरल एंड केमिकली स्टेबल। ये साख्त और केमिकल दोनों लिहाज से स्टेबल हो। इट मस्ट हैव द प्रॉपर्टी टू चेंज ड्यू टू म्यूटेशन। जिस तरह हमको मालूम है चेंज इन डीएनए इज नोन एज म्यूटेशन। The change may be either small or large, but when these properties are takes place, so it is the property of a genetic material. And be expressed in the form of Mendelian character, that each character is controlled by two alleles, and alleles are alternate form of genes. So although DNA is the genetic material in most organisms, and some viruses, RNA is the genetic material in plants, fungi, protists, animals, as well as in human beings. The genetic material is DNA, but there are certain viruses in which the genetic material is RNA. So according to study or according to the evolutionary point of view, RNA was the first genetic material. हमारे साथ RNA सबसे पहले विरासती मादा था जो इवोल्यूशन वक्त गुजरने के साथ साथ इसमें कुछ तब्दीली पैदा हो गई और RNA हमारे साथ अनस्टेबल था और इससे DNA मार्ज वजूद में आया। So DNA evolved from RNA with chemical modification and it is more stable and carry more genetic information. DNA जब हमारे साथ बन गया तो ये RNA से more genetic information इसमें बहुत ज़्यादा था और ये स्टेबल भी था। So first experiment जो DNA as a hereditary material के बारे में जिसने किया था उसका नाम था Frederick Griffith. Frederick Griffith performed his experiment in 1928. He was working with a bacteria known as Streptococcus pneumoniae, which cause a disease known as pneumonia. And pneumonia means inflammation of lungs. So there are two strains of bacteria, or we can say that there are two types of bacteria. R strain R for rough colony and S strain S for smooth colony when they culture on plate or on petri dishes. So the R type of bacteria they produce rough colony and S type of bacteria produce smooth colony. S strain bacteria have polysaccharide coat which give rise to smooth and shiny colony. The S strain is virulent, it means that they are pathogenic and it produce the disease pneumonia which produce death of an organism. And the R strain lack of polysaccharide coat, it give rough colony. R strain is non-virulent, non-pathogenic, it does not produce the disease and the organism will be live. So he performed the following experiment with these bacteria and saw the different observation. Or we can say that he performed four types of experiment. First of all, he injected S strain into the mice. Mice developed the pneumonia and die. Then in the second experiment, he injected the R strain into the mice live. In third experiment, he killed the S type of bacteria with heat. So heat killed S type of strain bacteria when enter into the mice live. Griffith found that heating kills the bacteria. In fourth experiment, he mixed the heat killed S type of bacteria with live R type of bacteria. Inject into mice and the mice become a die. All of these four experiments can be easily concluded with the help of a diagram. So rough strain non-virulent, the mice is live. Here smooth strain which is virulent, here the mice is a die. Heat killed, the mice is a live. When R strain live, they mixed with the heat killed smooth type, so the mice get the disease and it become die. स्पोर्ट नंबर एक्सपेरिमेंट को फिर दूसरे साइंसदानों ने ट्रांसफॉर्मिंग प्रिंसिपल के जरिए से कंक्लूड किया कि इसमें डीएनए हेरिडिटरी मटेरियल है जो कि ट्रांसफॉर्मिंग प्रिंसिपल का जिम्मेदार है, और तीनों साइंसदान का नाम है मैक्लीड, मैककार्टे and Avery. So conclusion of experiment. So he concluded that R strain had been transformed by the heat killed S strain. When the Griffith produce heat killed S strain, so the S type of bacteria can be converted, their DNA is converted into pieces. So the piece of the DNA which is responsible to produce the lipopolysaccharide, or we can say coat, it is absorbed by the Lyo R type of bacteria, and the R type of bacteria is transformed or changed into S type of bacteria. It produces the pneumonia in the mice, and the mice become die. So this happened due to the transfer of a transforming principle from the S strain to the R strain, which made the R strain virulent, pathogenic. It produced the disease. Although significant, his observation did not identify the biochemical nature of the transforming principle. Griffith in his life does not discover the transforming principle. These are the three scientists, Avery, MacLeod, and McCarthy, that they basically identified the transforming principle. They determine the biochemical nature of the transforming principle identified by Griffith. So these people purified DNA, RNA, and protein from the heat killed S strain and determined which macromolecule converted the R strain into the S strain. So in his experiment, firstly they treated heat killed S strain to proteases to digest their protein, RNases to digest their RNA, and then to DNases to digest their DNA, or we can say to break down or disintegrate protein, RNA, and DNA respectively. In their observation, he observed that treated with DNases inhibit the transformation of R strain. When he used the proteases, the transforming principle still takes place. When he uses the RNAs to digest all of the RNA, still the transforming principle is present. But when he apply the DNases enzyme, convert the DNA into pieces, then the transforming principle is stop. So proteases and RNases does not affect the transformation of R strain to virulent form to pathogenic form. So conclusion, they concluded that genetic material is DNA, not RNA or protein. But this theory was not accepted by biologists. So another two scientists, Hershey and Chase, performed his experiment in 1952, proved that DNA is a genetic material by his experiment. His postulate of the experiment are following that they use bacteriophage to experiment as follows. Bacteriophage is a type of virus that eat bacteria, or we can say that they attacks or infects bacteria. Some some viruses, or we can say bacteriophage, grow on a medium containing radioactive phosphorus, and some grow on radioactive sulfur containing a medium. Viruses have radioactive DNA that grown on phosphorus containing medium. Because DNA contains phosphorus but protein does not contain phosphorus. In contrast, virus containing radioactive protein which grown on sulfur containing medium, because protein contains sulfur and DNA does not contain sulfur. So these radioactive bacteriophages were allowed to infect bacterial cell and transfer their radioactive DNA or protein. That the bacteria either transfer their DNA, radioactive DNA or protein. After the infection, viral coat or particles were removed from the bacteria by the centrifugation process. This is the overall summary of the experiment in diagrammatic form. Sulfur labeled protein capsule are present in red color. So when this bacteriophage inject their DNA into the bacteria, so the bacteria they replicate their DNA into two, and then after centrifugation, no sulfur is present in the cell. So we can say that sulfur is not present within the DNA molecule. In the other set of experiment, phosphorus labeled DNA, radioactive form of phosphorus. So when this bacteriophage inject their DNA into the cell, this page is known as infection. Then the virus replicate their DNA into two. After centrifugation, phosphorus is present within the cell or present within the DNA. So from this experiment, He observed that bacteria were radioactive, which infected with viruses containing radioactive DNA, while bacteria were non-radioactive, which infected with viruses containing radioactive protein. Because protein are present in their capsid, and only the bacteriophage injects their DNA. So the DNA containing the radioactive form of phosphorus is present within the cell, are present within the DNA. So Hershey and Chase experiment conclude that DNA is the genetic material transferred from virus to bacteria, and protein is not responsible for heredity. RNA and protein. DNA is the genetic material for the majority of organisms. Protists, fungi, plants, animals, and human beings. The hereditary material is the DNA. But there are certain viruses that use RNA as their genetic material, such as SARS, severe acute respiratory syndrome, influenza, hepatitis C, polio, and retroviruses like HIV/AIDS. They all have either single or double-stranded RNA, and they use to replicate RNA in their life cycle, and their hereditary material is RNA. Prions are infectious agents that transmit their characteristics via proteins. Infectious protein particle is known as prion. If you like the video, please share, subscribe,